Welcome back to AP Biology, helping you study the study of life.
Hi, and welcome back to AP Biology, helping you study the study of life. Today, we're going to talk about protein synthesis. So, protein synthesis is the process of building proteins. And proteins are used for a lot of things in our bodies. They're used for enzymes and antibodies and some hormones. And we get the instructions for building proteins from our DNA. So, in short, during this process, DNA is transcribed into mRNA, which is then translated into amino acids, which is then used to build our proteins. Okay, so let's start with the first step, which is transcription. Transcription occurs within the nucleus of the cell. And in this process, we're going to transcribe the DNA strand into mRNA. And the M in mRNA just stands for messenger. And what happens is RNA polymerase attaches to the TATA box, which is just an attaching spot in the DNA sequence. And it begins reading the DNA strand and creating a strand that is corresponding but made out of RNA. So guanine will match with cytosine, cytosine with guanine, adenine to thymine, and thymine to uracil. And note that there is no thymine in DNA, so when there's an adenine in the DNA sequence, it will be transcribed into uracil instead of thymine, like it would usually be in DNA. And so as you can see here, we have our original DNA strand, TTAGCGTAGCGATG. And you can see how when we transcribe it, we change it into mRNA. Okay, so after the DNA is transcribed into mRNA, it will leave the nucleus and travel to the ribosomes through the cytoplasm. But first, before it leaves the nucleus, we have to edit it a little bit. Within DNA, there are exons and there are introns. Introns are junk DNA. It's unnecessary, and scientists do not know exactly why we have them in the first place, but we end up cutting out our introns, and this leaves gaps in our DNA strand. And so to connect the strands of exons, which is the coding part of the DNA, it's important, spliceosomes will, in a sense, splice together or stick together the strands. Okay, so we're almost ready, but first we are going to add a poly A tail to the 3' end and a GTP cap to the 5' end. And once we've done that, we are ready to move on. And the next step is translation. And this is when messenger RNA is translated into amino acids. And so this occurs in the ribosomes. And so here is a ribosome. It is made out of a large subunit and a small subunit. And the mRNA is read in sets of 3s. And these sets of 3s are called codons. And each set of 3 codes for a very specific amino acid. And we can use charts like this one to figure out what codon will code for what certain amino acid. So let's say that we have the codon GAC. So using this chart, we can see that the first letter is G, and we'll start in the very middle, the innermost circle. Then we will follow it to the A in the second ring, and then again to the C in the third ring, which leads us to ASP, which is our amino acid. So what happens during translation is the mRNA strand will go right between the 2 subunits. And what happens is tRNA which stands for transfer RNA, will bring the matching amino acid to the codon. And then the strand of mRNA will move, and the next codon is read, and the matching amino acid is brought. And eventually what we get is this just strand of amino acids that are held together by peptide bonds at the top of the ribosome. And they're all going to bind to each other, and the tRNAs will all be sent back into the cytoplasm to be reused. And this process ends when we reach what is called a stop codon. And this basically tells the ribosome, hey, I'm all done. I have translated everything that needs to be translated into amino acids. And the amino acids will build a very specific protein that will have a specific function. So any change in amino acids will change the structure and function of the protein. And these proteins will eventually be packaged and delivered out by cells, by other organelles within the cell. Okay, so to review, protein synthesis is the building of proteins. In transcription, the DNA is transcribed into mRNA. The mRNA will then go through some editing, cutting out the introns or the junk DNA and splicing together the exons, which is the coding DNA. Next, mRNA will be translated into amino acids within the ribosomes. And finally, these amino acids will build the proteins when they connect by peptide bonds. Well, that is all we have for today, folks. I hope you found this video helpful, and thank you for tuning in. Go to our channel for more videos that will help you study the study of life.